Hi there, students. Here's Unit 4. It's about DNA. I'm coming at you from a Saturday in my living room. So, this is pretty cool. I love this technology. So, in order to do well on the test, you should watch this video somewhere quiet, even in the center with headphones on, and really pay attention because it's not for nothing, right? All of this is on the test. Alright, so here we go. What is DNA? So, DNA is, it was partly discovered by this scientist, a lady scientist named Rosalind Franklin. She was a crystallographer. That's kind of an interesting job. So she worked with X-rays way back in the day. And she actually was the first person to take a picture of DNA. The picture here of where it says, what does it look like to you? I wonder if you think, hey, that looks a little bit like an X. Do you see the X? Kind of looks a little bit like an X to me. So she's the first person that took a picture of DNA. Now, people that really get the credit, though, are these two guys named Watson and Crick. Maybe you remember hearing about them. They took her work and they built upon it, and they figured out that DNA is a double helix. That's something that nobody else knew. And when we say double helix, it pretty much means like a twisted ladder. And they knew that there was something on each side of the ladder, and then there were kind of like rungs to the ladder. They built this. This is a picture here. They built a wood model of DNA. That was a really big deal. Nobody knew about DNA. I mean, nowadays it's like, dime a dozen about DNA. You watch Jerry Springer, or I don't even know if he's still on. What's his name? Maury Povich. You watch Maury Povich and it's like, oh yeah, I want a DNA test for my baby. And they say, you're not the father, or whatever. That's DNA, but it's actually a really relatively new science. It's pretty amazing. So now that we know what DNA is, we know that it's a double helix. What is it made up of? So, DNA is a polymer. Poly means, what's that? Many? You're right. Poly means many. So it's a polymer, see poly, many, that is made of monomers. Mono means, yes, that's right, one. DNA is a polymer made of monomers called nucleotides. I wonder if that sounds familiar to you, nucleotides. I hope so, because we learned that in the very first unit when we were learning about organic molecules. Now, DNA is a double helix like we said, and it has these nucleotides. It's these specific ones. One's called adenine, one's thymine, guanine, and cytosine. Now, these pair together. See how they're kind of paired together here? Those, they have to follow certain rules. They can't ever, an A can't ever get with a C, a G can't ever get with a T. It's always got to be A, T, C, G. Maybe you remember when I was singing my Katy Perry song, C, G, A, T, C, G, A, T. That's what I was talking about, C, G, A, T. So they have to pair. There's no rule of life. So here's a great way you can remember it, besides me chanting C-G-A-T as Katy Perry. All tigers can growl. That is going to be a great thing for you to remember. All tigers can growl. You are most definitely going to have to use this. So on your notes where it says all, and then tigers can growl, next to all you should write adenine. Next to tigers you should write thymine, and so on. So write those in there. Now, there is no other way. It will always, always, always be true. Now let's do this base pairing practice here. Whoops. Oh wait, technical difficulties. Alright, there we go. Let's do this base pairing practice there on the side. You're going to have to do this on the test. You're going to do this in class. It's a big, big part of this unit. So we have something here that says A T, C, G, G, T, C, A. Somebody needs to figure out how to write better on this. All right, now I'm going to do the base pairings. So, whenever I see a T or an A, I'll put a T. Whenever I see a T, I'll put an A. Whenever I see a C, I'll put a G. Whenever I see a G, I'll put a C. G with C, T with A, C with G, A with T. That's how you translate. That's how you base pair. Now, I want you guys to try the next one, the next 3 on your own. You can push pause on me. Go ahead and try that. That's going to be real important. Okay, so you should have just tried that. You will try these. This doesn't matter anymore. Okay, hello. Central dogma. So when you hear the word dogma, it basically means dogma. It means belief. Hey, did I spell that right? I don't know. It means belief. So the central belief of molecular biology is that DNA flows in one direction, and it's always going to flow in the same direction. It's going to go from replication to transcription to translation. Whoa, what the heck did I just say? Don't worry. We're going to talk about it more. So you can see there in your first blank, information goes from DNA to RNA to proteins. So DNA was all tigers can growl. Then it gets translated to RNA and finally into proteins. We're going to talk about those next 2 steps now. That one. Now, DNA replication. Do you remember what stage the cell cycle is DNA replicated? Oh, what's that I hear everybody saying? Let's see. It's the synthesis phase. Did you remember that with mitosis? Remember the cell cycle, and the cell spends most of its time in interphase, and during the S phase, the DNA is copied. Now, during the replication process, this is going to get really... This is going to get super confusing, and don't worry, we don't need to really know about the total minutia. Minutia means all the little tiny details of this, but enzymes, so they unzip because it's like a ladder, right? So it's like they take off one of the sides of the ladders. Now, what's going to happen is that 2 new strands of DNA are going to be made, where one is going to be made from the original and one new. That process, it's called semi-conservative. See that word down there? Semi means sort of or partly. Conservative means saved. Means saved part of it. Now, remember again, now this is where the base pairing rules really come into play. That if we're pairing things off and we're taking things apart and unzipping it, T has to know that it pairs with A. C has to know that it pairs with G. Otherwise, it's going to be total chaos. And remember, our DNA is what makes us us. It's what tells our cells what to do. It's in every single solitary one of our cells. So it's a really big deal for it to know where it's going. Now, so we talked about DNA, so now I'm going to throw a new thing at you called RNA. So transcription converts DNA into RNA. Then the RNA is translated into a string of amino acids, and they are called proteins. Now, I like to tell my students that I think of this as maybe, I don't know if you guys have ever seen the TV show Mad Men, but it's a TV show. It takes place in like the 50s and 60s. And back in the day, all of like the fancy businessmen had secretaries. And what they would do is if they wanted to write a letter to somebody, because this is before email, they would call their secretary in and they would say, "All right, Betsy, I need you to take a letter." So Betsy, the secretary, would sit down with a notebook and she would write what he's saying in shorthand. Shorthand is like a special language for secretaries where they can write a whole bunch of stuff really fast. It would be similar to a court reporter. If you've ever seen like a TV show with a court reporter, she's using a shorthand. So this secretary would write out in shorthand. She would transcribe what he is saying, but it would look like gibberish on her paper. Then when he was done with the letter, she would go out and she would go to her typewriter and she would translate that letter into something that makes sense. So it's kind of like what DNA, RNA, proteins is. DNA is the guy talking, RNA is the transcribing, the writing it down, and translation is when it's turning into protein. So hopefully that helped you understand that part. Now, how does DNA make RNA? It's called transcription. Oh, hey, Mr. Parr has a song for that. So if you didn't already listen to your song, then you can push pause on me and watch the song, or you can always just fill in the song later. But you definitely have to watch the song because that's quite a bit of your pages of your packet. So, all right. DNA versus RNA. So this is like one of my favorite things. If you want a prom date and you're smart, maybe you'll use one of these pickup lines here. See the cat? If I had a choice between DNA and RNA, I'd choose RNA because it has U in it. Or I wish I was adenine, then I could get paired with U. Wait a minute, U, that's not one of our letters. That's a letter that's in RNA. So DNA has deoxyribose, and now RNA has the sugar ribose. So those are two different kinds of sugars. Anytime something ends in OSE, it means sugar. Now, DNA has A, T, C, and G, or CGAT, or however you remember it. Now, RNA has something a little bit different. It still has the A, still has the C, still has the G. It just has a U instead of a T. That's why those pickup lines make so much sense. RNA is double strands, and it contains instructions. RNA is single strands, and it also carries instructions. So those are some big differences there between DNA and RNA. Now, like I was telling you about with the secretary, when she's transcribing, she is writing word for word what her boss is telling her to write in the letter. She's not making up anything because that's her boss. He wants the letter how he wants the letter. So it's an exact copy. It's important that it's an exact copy because our DNA is exactly what makes us us, and if it was off, we would have big problems. Now, you can see here, this is a good visual here, where you can see DNA is double strand, RNA is single strand. That would be an example of transcription. From transcription, we'll methylation. Now, this is the process where mRNA is transferred into a polypeptide. I gotta say, I geek out over that word. That is like my favorite word in science, polypeptide. So, mRNA. There's 3 types of RNA, and mRNA, the M stands for message, messenger RNA. Oh, got a little ahead of myself. Here we go. So, mRNA, messenger RNA, tells the cell what type of protein to make. It's the message, it's the instructions. rRNA, ribosomal RNA. Do you remember that there's something called ribosomes? So that's where the energy is going to be, the proteins. Ribosomes are where proteins are made. tRNA, transfer RNA, it carries the amino acid from the cytoplasm to the ribosome. I always remember, for some reason, I always remember tRNA as, whoa, sorry, I hope that didn't make you dizzy. That's what I was trying to do. I always remember tRNA, I like to draw this little visual of a little cart, and it has wheels on it, and it says tRNA, and this is amino acids. I don't know if that helps you, but then you can remember that it carries amino acids from the cytoplasm to the ribosomes. So, how can we remember all of this? I know. I pity the fool that makes proteins. I don't even know if you guys get that. I don't know if you know Mr. T. That might be kind of too 80s for you. Anyway, Mr. T makes proteins. M for messenger, R for ribosomal, T for transfer. So, Mr. T wants you to remember how to do translation. It's pretty clever if you can remember Mr. T. So when we're talking about amino acids means proteins. Proteins are what tell our bodies what to do, what to look like, how to act. We're made of proteins, basically. So translation is going to convert the mRNA into a polypeptide, which is a protein. Now it has to be read as 3, so every 3 letters equals a protein. It's called a codon. Remember when I was singing That's DNA? Amino acids, 3 base pairs make up a codon. Amino acids get turned on. That's DNA. Remember that? So a codon is a sequence of 3 nucleotides that form an amino acid. So they always have to come in 3s. There's no exception to that rule. I feel like this unit talks about rules a lot. There's so many rules for all of these things. Now, start codons signal the start. Stop signal the stop. Wow, imagine that. So there's little 3 letters that mean start. There's 3 letters that mean stop. Don't worry, this might be feeling like your head is about to explode, but we're going to do some examples of this in class. We're going to look at this, and I think it will help. Now, there's this little chart here, and it is made of the 20 amino acids that we are made of. So we are made of 20 amino acids. Now, this is the chart here, and it is showing us which amino acids are made by which letters. We're going to do an activity in class where you're going to actually use this chart in a sense. A long string of amino acids make a polypeptide chain, which is a protein. So, all of this translating and transcribing and whatnot, what if I make a mistake? Because, you know, nobody's perfect, right? It's going to happen. It's bound to happen, and that's okay. So, sometimes there's information out here. You know, I mean, the internet is full of examples of things where people are just making crap up, right? So, here's an example of something that somebody made up on the internet, and you can see that they used Photoshop and they made it look like this was real, right? But obviously not real. But let's look at some real animals that you would think, hey, that's not real. What's wrong with this picture? Let's see. 1, 2, 3, 4. Wait, why is that one over there? Why is that looking like that? And then wait, doesn't he still have froggy legs there too? That has a lot of extra legs. That's real. That's caused by a mutation in his gene. Here's a cat. I don't know if you like cats or not, but I especially don't like these kind. Yuck. But that's real. Dolphins, on the other hand, are my favorite. So there's actually such a thing as pink dolphins. This is one, and it's caused by a mutation. Here we have a white, I don't know if that's an alligator or a crocodile. I always forget. I think it's an alligator. No, crocodile. I think it's a crocodile. And it has no pigment. It's not green. Isn't it supposed to be kind of like that ugly green color? Yeah, it totally is, but it's not. So that is an example of a mutation as well. Okay, so now wait a minute. How could I show you a picture of a person with a mutation? We all have mutations in our genes. They just usually get caught. This kid has Down syndrome, and remember we talked about that, was where he has an extra chromosome on his 21st. And that's an example of a mutation. So like I said, in reality, I mean, mutations are everywhere, and thankfully they're there because that's what creates genetic diversity. And remember, without genetic diversity, things would be pretty boring. And also, we would have too many recessive genes and we wouldn't survive. So mutations can be a pretty good thing in a lot of cases. Oh, Mr. Parr, here's another song. If you would like to listen to it, you can. Otherwise, I don't have it on your notes, so it's optional song. Now, a mutagen. A mutagen is something in the environment that causes a mutation. So this is real. This is something that could really happen. Ultraviolet radiation from sunlight. Okay, so if you go out in the sun for too long, you may get a sunburn. You may get some moles. Those are mutations that happen. Chemical pollution. So maybe you know about the Joker from Batman. He fell into a vat of chemical waste, and he then became yucky. And so that is an example of a mutagen. Now, there's a few types of gene mutation. Usually, they're caused when there's a problem in the transcription or translation phase. If something gets copied wrong, then the protein that it makes is not the right one. And it would be a kind of a problem because the proteins are what tell us what to look like or whatever. Now there's this really great thing called DNA polymerase, and it fixes everything. So when I say erase, it's called DNA polymerase. Let me show you here. Poly means many. See the word? Do you see a word hiding in there? It's like a little word search game. See that word erase? That can help you remember that it's like an eraser, and it helps to fix any errors that could be on some DNA or an RNA or whatever before it becomes a problem. Now, there's a couple types of mutations. One mutation is called a point mutation, and that's where just one little letter is off. Just one letter gets copied wrong. Now, it's not too big of a deal because usually DNA polymerase will find it, fix it, and it's not too big of a deal. Not gonna hurt anything. Also, you'll see that the same letters or different letters code for the same proteins. So a lot of times if it's just off by one letter, it's still going to call for the same protein. So there will be no effect in the phenotype, and you just wouldn't even know. We probably have this happening all the time and we don't even know. Now, point mutations are also called silent mutations. Now, it's because, like I said, it doesn't change the protein and it does not affect the phenotype. So that is important to know. You'll have to know a point mutation is also called a silent mutation. Here's another one, and it's called a frame shift mutation. Now this is when a letter is added or deleted to the sequence. Now this is going to cause some problemos. This is going to make everything totally wrong. So, for example, if you have something called, like, if you have a sentence, let's do the sentence, time to dream, right? Time to dream. That does sound good. I could go for a nap right now. Time to dream. Now, let's say that instead of that on accident, this E got deleted. Now remember that the proteins are coded for in codons. Codons are 3 letters. So if this is going to be wrong, then it's like still going to have 4 spots, then 2 spots, then 1, 2, 3, 4, 5. It's kind of like a fill in the blank. So it's going to start filling in the blank, and it's going to say, okay, T, I, M. And if this red one got deleted, then instead of putting the E, it's going to put the T. Okay, and then it's going to fill in with the rest of the letters. O, D, and then R, E, A, M. So see how there's kind of a problem there, because now it says timid odd ream. I really don't understand what that's trying to say. So if there is a frame shift mutation, it's going to be a really big deal. It doesn't make sense anymore. It's really important that DNA polymerase fixes things, fixes the mistakes. Otherwise, it's a big deal. So if there's a nucleotide inserted or deleted, it's a... See, here is the chart, and you can see here that a single letter change is not too big of a deal. So, see, tame to dream. Yeah, you know, it's not really super understandable, but kind of. Then here we have, if you reverse it, emit to dream. Maybe you could still sort of figure it out. But then here, when you delete Tito Dr. Eam, what? I don't understand that at all. Or how about this one? Tim et Orim? Who knows what they're trying to say? Doesn't make sense. All right, so now is question time. We're going to do the Translation Olympics when I see you guys. But here is the question. You need to email me right now so that I know you watched the video and you know I'm serious, right? All right, so you've got to email me. And what I want you to email me is, I want you to email me the cat ate the rat. Good thing, because I hate rats. There's your password. So I'll be seeing you guys soon. Make sure you finish these and make sure you watch Mr. Parr. Don't stress out because you'll all do great on the test just like you have been. So trust yourselves, do your homework, and see you in the next video. All right, bye.